Okay, so hello everybody, and this video lecture is covering chapter 11, but only physiology, really physiology of nervous tissue. Because anatomy of nervous tissue, that lecture you will find in the lab section, and there is another professor who recorded it, so you can watch his videos. Okay, so let's go ahead and begin. Now, the first thing I want to tell you is that this sentence, all cells establish electrical potential called the resting cell membrane potential 70 to 90 millivolts. And if you took any physics classes, that might make sense for you. If you didn't, that should have no sense, right? Because like what is this electrical potential? What is, what is this voltage? So for those students who didn't take any physics classes, if to put it simple, your cells, every single cell in your body is a little battery, like a battery over here. And what does it mean you have a battery? If you look at your battery, all batteries, they have positive and negative charge, right? So what we have separation of opposite electrical charges. And why would your battery has it? Because when you separate charges, you actually store potential energy. So this battery has potential energy. And this separation of charges is called electrical potential, because it has this potential energy, and it's called voltage. And voltage can be measured, right? So now your cells also like a tiny small battery. So if I draw a cell over here, like, so here's your cell, here's the nucleus, right? Actually what happened, right, so here's some organelles, but your cells are negative inside, negative, and positive outside, right? So that's why they're batteries, because your cells also separate ions, right? They separate those charges, and they became a little battery. Negative inside, positive outside, right? And this is what called resting membrane potential, because membrane is what separates those charges. So every single cell look like this battery, right? With separation of charges. Now, when those electrical charges start moving, like it shows over here, right? When charges are moving, we have electrical current. So some of your cells are capable of creating this electrical current. All your cells are batteries, but some cells also can create this electrical current. And those cells are muscle cells and nerve cells, right? Now, because we have separation of charges, we can measure this potential energy in voltage, and it will be minus 70, minus 90 millivolts. And we say minus because we're referring to this minus charge inside a cell, right? Now how does it happen? How your cells are capable of creating this potential energy? But to understand it, we need to first look at membrane proteins. So it says here, role of membrane ion channels in this establishing of this resting membrane potential, right? So the cell membrane is phospholipid bilayer, right? And this is what you study in your Bio 50A. And what we have, transmembrane proteins inside the cell membrane. And we have two types of this. Well, some of them are ion channels. So those proteins can have different functions, but some of those proteins that are integral proteins that are inside a cell membrane, they are ion channels. So we have two types of ion channels, leakage channels or non-gated. Well, leakage means something leaking right through this channel. So they are always open. Now what's leaking? Ions, right? So ions leaking from these leakage channels, and they always open. And another type of proteins are gated channels, right? So if over here I have like a cell, right? So here's a cell, again, nucleus. And for example, here's some protein. Of course, it's really huge, right? But this protein is a channel. So it's allow ions to move in and out. And some of them, they have gates, right? Like it can be inside, outside, or it can be on both sides, right? And when we have those gates, this channel doesn't stay open all the time. So they can be open, closed, right? And we call them gated channels. Now, how to open these channels, right? If you have a door, you know how to open the door, right? You either need to pull it or push it, or if it's locked, you need to use keys. Now, how to open those gates? Well, there are three ways how to open it. That's why gated channels, we have three types. They can be chemically gated or ligand gated. Then some chemical will attach some molecule, can be neurotransmitter, can be hormone. So some chemical will bind to this protein and gate will open, right? So it's called chemically gated. Some channels are voltage gated. Now they will be open when this voltage across a membrane changes. Remember, across a membrane, you always have -70 to -90, sometimes even -100. So when this voltage is changing, then gates can be open, or they can be mechanically gated. So this gate will open as a response to physical deformation of receptors. Like what physical deformation? For example, when somebody touches your skin, right? That's a physical pressure, right? That's some pressure applied to your skin, and this pressure can open those gates, right? It's by the way, if you ever ask yourself, how do I feel? Somebody touching me, how do I feel? Right? So how do I feel this touch? So the first channels that are involved in this process of you feeling that somebody touching you are mechanically gated channels. Because when somebody touches you, they disturb these little gates in your receptors, in neurons, right? And they open and, something happened. Lots of stuff happened after that we will discuss in this chapter, right? So again, ion channels can be leakage channels or gated channels. Oh, by the way, here's a picture we can see. So this one is ligand gated. So any chemical that attaches to a receptor called ligand. So you see this channel is closed right now, right? It doesn't have a gate, but it can have a gate as well. But it's closed. Now neurotransmitter attaches and channel is open, right? So that's a ligand gated. Now this one, look, that's inside of your cells, negative. Remember, your cells are really negative. Positive outside. So this channel is closed. Now, when we change the voltage, now look what happened. Now cell is positive inside and negative outside. So you see, we reverse this and channel is open. So this is voltage gated. It doesn't necessarily need to be complete reverse. It can just be less negative to open this channel, right? But we have change in voltage and channel open, so that's voltage gated. Okay, now when we know that we have two types of channels, leakage and gated, let's see how your cells create this resting membrane potential. Now, resting membrane potential is generated by two things. First, difference in ionic makeup in ICF and ECF. And ICF is intracellular fluid, so it's inside, and ECF is outside, right? So we have different ionic makeup. And this one factor that allow your cells to create this resting membrane potential. Remember, potential means separation of charges and a differential permeability of plasma membrane. Now, first, what different ionic makeup? So over here, you see a neuron, right? So here's the soma, and this is axon. And they're just looking at a little part of axon. And you can look at soma, at dendrites, doesn't matter. What you will see negative inside, positive outside, right? That's what's called resting membrane potential. And if you measure it, here's about minus 70 millivolts. Right now, what is different makeup? I'm going to show you on this picture. What the different makeup is that outside of a cell, you have high concentration of sodium. So lots of sodium in ECF, right? And inside the cell, right there inside, so right there, you have high concentration of potassium. So lots of potassium, right? So that's this differences in ionic makeup. So that's what it is. To remember it, you know, in physiology, we call our cells salty bananas. Salty, because imagine if you have banana and you put salt on top of it, right? So outside is salt, like sodium chloride. And by the way, we do have high concentration of chloride ions as well. And banana is rich in potassium. So inside we have lots of potassium, right? Now, another factor that generates this separation of the charges is differential permeability of the plasma membrane. Just to put it simple right now, just think that plasma membrane, over here, we have ion channels, is permeable to potassium. So it's permeable to potassium, but not sodium. Well, it does permeable to sodium, but way less sodium can move. So we have lots of these ion channels. So it's way more permeable to potassium. So just to make it simple, remember that plasma membrane is way more permeable to potassium through leakage channels. So we have lots of these leakage channels. So potassium can actually move out. Right, so that's a differential permeability of plasma membrane. That makes sense? I hope it does. Next. Okay, so here now we go in details. Differences in ionic makeup. So inside the cell, we have high concentration of potassium, right? And outside, high concentration of sodium. So you see over here, sodium outside, 150 milliequivalent per liter, and inside is only 15. But potassium inside is 150 and outside is 5, right? So that's our differences in ionic makeup. Now, differential permeability of membrane. Remember I told you that it's mostly permeable to potassium, right? So you see, I put the arrow over here, so potassium can move through the membrane. Can sodium move? Yes, but it's 75 times more permeable to potassium. So we can kind of ignore this movement of sodium. So we can just say like potassium has lots of leakage channels and potassium can move. Now it's also freely permeable to chloride. So because it's freely permeable and can move in and out, so we can also kind of ignore it. Now, if you look over here, it's impermeable to A minus, that means proteins. So inside the cell, we have lots of proteins, right? So here's a protein, here's a protein. And proteins, they are negatively charged, right? Okay, now, the question is, why do we have lots of sodium out, right? So why do we have lots of sodium out and why do we have lots of potassium in, right? So here's our question. Why do we have it? Well, the reason for that, I'm sorry, reason for your cells to be salty bananas, because all your cells, they have like a special protein over here, right? So it's a special protein that can attach over here. So it can attach sodium, 3 sodium atoms, 3, right? So they all attach over here. And 2 potassium from outside, right? And it can move this potassium in and sodium out, right? This special protein is called sodium potassium pump. And it's called pump because it uses ATP, right? So it uses ATP, so it's active transport. But what sodium potassium does, every time it moves sodium out, right, potassium in, sodium out, potassium in. So if you constantly moving sodium out and moving potassium in, what do you think gonna happen? Well, if you're moving sodium out, then concentration of sodium outside gonna increase. If you're moving potassium constantly in, right, you will have more and more potassium in, right? So that concentration increases as well, right? Now, sodium potassium pump creates concentration gradient. So when we have this difference in concentration in and out, like lots of sodium out, very little in, it's called concentration gradient. So sodium potassium pump create concentration gradient for both sodium and potassium, right? Now, well, potassium, remember, potassium can move out. So potassium can move out. Why potassium would move out? Well, because any molecules and ions and, you know, any substances, they will move down their concentration gradient. So if we have lots of potassium here and very small amount outside, potassium will move out, right? Why it will move out? Because I told you, remember, we have lots of leakage channels. So potassium will move out. Sodium, of course, would love to go back, but we don't have that many channels. So it's pretty, you know, it stays mostly out of a cell, right? So now when potassium moving out, right, so more and more potassium moving out, and potassium is positively charged, right? So what do you think gonna happen with your cell inside? It became more and more negative, right? So negative, because we're removing positive ions from inside a cell, so cells became negative inside. And this is called electrical gradient, right? Now, and that's our potential, right? This is our resting membrane potential. Now we separated positive and negative charges. And we separated it first by creating chemical gradient, concentration gradient, and then, they moving out, well, and electrical gradient kind of, like if electrical gradient tried to push it back in, so when concentration gradient equals electrical gradient, then we have equilibrium, and that equilibrium we call resting membrane potential. Right, so the equilibrium potential or resting membrane potential that has developed is called resting cell membrane potential, right? So again, we sodium potassium pump separates sodium and potassium, creating concentration gradient. Potassium start moving out, down its concentration, but potassium also wanna come back in. Why? Because it's positively charged attract to negative charges inside, because of electrical gradient. And when we have an equilibrium, we call it resting membrane potential. Right? Okay. So I hope that makes sense. Okay, let me stop, pause record. Okay, so here we get to this point, right? So now we know that all our cells create this resting membrane potential, minus 70, minus 90, minus 100 millivolts. Now, can it ever be changed? Actually, yes, it can be changed. So membrane potential can be changed. When? Well, because to create this potential, we use separation of ions, right? And concentration of sodium and potassium inside and out. So if we change this concentration, we're changing membrane potential. Also, we create resting potential because of different permeability of membrane, right? So if we change the permeability, then we change our membrane potential. Now, changes in the membrane potential does not happen in every cell. It only happens in muscles and nerves. And these changes are actually signals. They use the signals to receive, integrate, and send information. That's very interesting. Now I want to show you another slide before we get here. Okay, so let's just look at this diagram. Now, where we at? This is neuron, and this part of neuron is called dendrite, right? So here's a dendrite, and they take a little part of dendrite. Now, this neuron is resting, so it's really doing nothing right now. It's just resting. When it's resting, it has membrane potential, resting potential, negative inside, positive outside, right? And if we measure it, it's minus 70, right? So here's our resting potential, minus 70. So it can stay like this for days and weeks and I don't know, months if you don't use this neuron. But if we changing this voltage, right? And see what happened over here. We go from minus 70 to minus 69, 68, 65. So we're moving towards zero. Then this is called depolarization. So this neuron is polarized. It's polar, plus minus, right? So it's polarized. Now, when we start moving towards zero, we depolarize it, right? So it's called depolarization. Whenever this voltage start moving towards zero, right? Now, on this picture, we in the same spot, right? This is a dendrite. So again, minus 70, resting potential. But this time, it's changed, but it changed to more negative even, right? Minus 70, minus 71, minus 72. Because neuron is polarized, now this is hyperpolarization, right? So this is how resting potential can be changed. So it either can be less negative, and then it's called depolarization, or it can become more negative, and this is called hyperpolarization, right? Okay. Now, remember I told you whenever we change this voltage, right? Here or here, doesn't matter. Whenever this voltage is changing, your cells can use it as signal. Now, we have two types of signal. Graded potentials and action potential. Now, remember, resting potential is not a signal. It just like you bought a battery and you have this battery, it's doing nothing. But when you put this battery in your flashlight, then, you know, you use this potential energy to do something, right? The same with your cells. And now we're going to talk about neurons specifically. So your resting neurons, they just those batteries that they do have separation of charges. They have this voltage, right? Resting potential. Now, when we start changing it, it's either graded or action potential. Now, graded potential shown over here in green line, right? And graded potential actually happens in dendrites and soma. All right, so see, we're moving towards zero, right? And if we're moving towards zero, this will be depolarization. Now, graded potential are short distance signals of dendrites and somas. Now, what's shown over here in purple line, this is how action potential look like. And this is long distance signals. And if you're talking about neurons, that happen in axons, right? So two type of signals, short distance kind of signal and long distance signals. And this is how they look on a graph. So here you can read that graded potentials, they are short-lived, localized changes in membrane potential. It can be depolarization or hyperpolarization, right? So we can go towards zero or away from zero. It's still graded potential. And graded potentials, they spread as local currents, change the membrane potential of adjacent region. So here we are in a dendrite. So your resting dendrite is positive outside, negative inside. Now, if some stimulus is applied, and again, stimulus can be, somebody touching you, right? Or you even hearing me. That's the same. It's a stimulus to your neurons. Now, when stimulus is applied to dendrites or soma, you see how we're changing our polarity. We became kind of positive or less negative. Well, in general, it gets less negative. It's not positive completely yet, right? But it gets less negative. So we're moving toward zero, right? This is depolarization. And it just kind of moves in all directions, right? Because those positive charges, they just diffuse. That's a graded potential. So graded potential, okay, so I just told you that this is graded potential, right? But what exactly happens over here, right? Why we have positive inside? So when stimulus is applied, so let's imagine that's your arm, somebody touching your arm with a needle maybe, right? But slightly, not too bad, right? Somebody touching with a needle. Now, you have like pain receptors in your skin. And what you have in those dendrites, you have channels, ion channels. And those ion channels are gated. So when you apply this stimulus, this channel is mechanically gated. So when you apply the stimulus, this gate opens, right? So here's, I put it right here. The gate opens, right? And what moves in? Sodium. So sodium, sorry for my writing, start moving in, right? So here more sodium. Sodium moves in. And sodium is positively charged, right? So this plus, it's actually not just some, magic plus. That's a sodium ion start moving in and giving us this graded potential, right? So it's what it says over here. Graded potential occur when a stimulus causes a gated ion channel to open, right? It's not always sodium, but in most cases, it's sodium. Graded potential can be receptor potential, generator potential, postsynaptic potential. Don't worry about it right now. It will be explained later. But we have different type of graded potential. Now, magnitude of this graded potential directly proportional to the stimulus strength. So that means if you have weak stimulus, just few sodium moves in. If you have strong stimulus, you have more sodium moving in, and it means you increase the amplitude, right? So magnitude. So magnitude increases when stimulus increases. And it's also, with distance, it gets weaker and weaker and weaker, right? So it's kind of like where you apply stimulus, you have, you know, very strong change, and then in voltage, and then it just pretty much disappear away from the stimulus over here. Now, what is action potential? Okay, let's look over here. Let's see what picture I want to show you. So now graded potential, when we're talking about neurons, happens in dendrites and soma. Action potential happens in axons, right? Oh, I can look over here. So if you look over here, so here's our neuron resting -70, right? Now, well, the voltage is not changing just a little bit, it's changing drastically. It's actually from negative became positive. So if you have like your axon like this, right? And remember, resting is, I don't know why I'm making those tubes all the time, but negative inside, positive outside, right? So action potential give you complete reverse. So you're gonna have positive and negative, right? So that will be reverse of this polarity with from -70 to +30, right? And that's what action potential is. So that's a big picture here. Resting -70. Now, neuron is polar over here, polarized. Now this one is depolarization because we're moving towards 0 and even to positive. And now we're moving back to negative, repolarization. And when we below -70, this is called hyperpolarization because it's even more polar, right? And back to resting. So resting, depolarization, repolarization, and hyperpolarization. Okay, now in a resting state, just remember, in a resting state, only leakage channels are open, right? All gated channels are closed. Okay, now we have, to generate action potential, we have initiation. Now let's look over here. So here's your neuron. So those are dendrites, this is soma, this is axon. Remember, action potential can happen in axon. Now we apply stimulus to soma or to dendrites because dendrites are receiving region. So when stimulus is applied, what happens here is graded potential. Now this graded potential, remember, it decreases in amplitude. So it's actually get the electrical current, so this is electrical current, get weaker and weaker, right? So it's strong current and then it decreases. So you can see over here, when we just in this area where stimulus is applied, we have like almost -40. But when it moves further and it reaches the beginning of axon, it's called trigger zone, it's below -55. So this line is called threshold. So it's below the threshold. And if it's below threshold, we do not have any action potential. So no action potential if stimulus is not strong enough. Now here stimulus is strong enough that by the time it reaches this trigger zone, it's still above the threshold and action potential is generated. So the first step in generation of action potential is initiation. And it's arrival of graded potential with sufficient strength to stimulate action potential. So no initiation here, we do have initiation over here. Now, what would be example of this? So imagine that you are you camping somewhere, right? Outside. And it happened with me many times, and I'm sure it happened with you. So you're sitting, and then all of a sudden, somewhere next to your knee, you see an ant. So the ant was crawling all the way from my toes, right? All the way up to my knee, and I didn't feel it. I only feel it when it gets to my knee, right? So that ant on my leg, when it's walking on my skin, it actually stimulates my neurons. But stimulus is so weak that I cannot feel it because action potential is not generated and it's not reaching my brain. But when, when ant get to my knee, I have maybe, more sensitive neurons, or that stimulus is enough to initiate action potential and I feel it, right? The same with mosquitoes. How many times you sitting and mosquito is sitting on your skin, you don't feel it, right? Because it's sitting, it's actually disturbing your neurons, but it's so weak stimulus that you don't feel it. But when mosquito bites you, increases the stimulus, right? Then action potential is generated and it reaches your brain and you're like, oh, I feel mosquito bite, right? This is what initiation of action potential. Because all the sensory and motor neurons, they work by creating those action potential. Now, next step is depolarization. Okay, so I'm going right here because I know you can read those slides yourself. So this is our resting potential. Now we have initiation. So we have stimulus strong enough, right, to generate action potential. Now, when we at resting stage, only leakage channels are open. All gated channels are closed. Now, if you have stimulus strong enough, something gonna happen inside your axon. And look what's gonna happen over here. Sodium gated, voltage gated channels are open. So sodium start moving in. When sodium positively charged start moving in, obviously you are negative inside, right? Now positive ions moving in, cells became less and less negative, right? So that's what this line is. So this is called depolarization because we're moving towards zero, depolarization. So sodium channels are open. Now somewhere in around this area, potassium channels are open, right? So this, but we still have lots of sodium in, but look, we start losing potassium. Now, somewhere at the peak of this depolarization, sodium channels now closed and potassium stay open. So now we start losing potassium. So we were gaining positive charges, so we became positive. Now we're losing positive charges, so we're going back to our minus 70, right? So sodium are closed, potassium is still open. Now we're losing potassium, so we depolarization, right? So what happened over here? Sodium, so remember, that's your normal cell, right? What we have? Negative inside, positive outside, and we have lots of sodium and lots of potassium in. Oh, that's not potassium, potassium. So we have sodium out, potassium in. Well, now what happened here? Right there, look, sodium in. So sodium in, and potassium out. So if you have sodium in, potassium out, you will have sodium inside, right? That's a cell. And potassium outside, right? So when we get to this point, our voltage is okay, negative again inside, but now we have sodium in, potassium out. That's not right because we need to be salty bananas. So here where we have this sodium potassium pump, right? So here we start using sodium out, potassium in with ATP with sodium potassium pump. So it shows over here, sodium out, potassium in, sodium out, potassium in, right? But because these potassium channels, they stay open a little bit longer, that, then we have this hyperpolarization. So resting potential, depolarization, repolarization, hyperpolarization, and back to resting potential, right? So basically, to put it simple, your neurons use movement of sodium and potassium in and out to create electrical current. And we call it this electrical current action potential. Okay. Okay, so here we can look at the picture, how it happens. So where we at? We are at very beginning of axon, right? This is called trigger zone. So stimulus is applied somewhere over here where dendrites is. So dendrites and soma, they create graded potential. Now graded potential reaches this trigger zone. And what happened, it's open this sodium voltage gated channels. Sodium start moving in, right? So here sodium start moving in, and of course it diffuses in both sides, but if it diffuses backwards, so kind of our electricity start going back and forth, and that's not good, right? Because we want an electrical current only go along axon to axon terminal. We don't want just electricity moving back and forth. But in this area, we're still losing potassium, right? We're still losing potassium. This area is in a refractory period. So that's why, this just dies off. So electrical current action potential always moves in one direction, only along axon, right? It doesn't move back, because if it moves back, these positive charges, we're still losing them because potassium channels are still open, right? Makes sense? Okay. So very quick, what time is it? Oh my goodness. So very quick recall. So for us to generate action potential for other neurons, we need first to generate graded potential in dendrites and somas. And if stimulus is strong enough, then we have action potential, right here. If stimulus is not strong enough, no action potential. That's why we have different type of stimuli, right? We either have sub-threshold stimulus, those are weak, and it will not cause action potential, or threshold stimulus that is strong and it will cause action potential. So if you go back to this diagram, right, this stimulus is sub-threshold stimulus. So it not generating action potential. And this stimulus is threshold. And threshold means by the time this electrical current graded potential reaches trigger zone, it's above minus 55 millivolts, right? If it's above, then action potential is generated. Okay. Now, all action potentials are alike, and they are independent of stimulus intensity. Remember, I told you that graded potential, it depends on how strong the stimulus is. Action potentials are all the same, from minus 70 to plus 30. That's it. It cannot be more or less, right? Now, if all action, and your brain receives this action potential, and this is how your brain interpreted what kind of stimulus you have. But some stimuli are strong and some are weak. So if my brain receives the same action potential, how my brain can determine or determine the difference between weak and strong stimulus, right? So if somebody touches you very lightly, right, or somebody punches you, that's a different stimulus, right? I hope nobody punches you, but just let's pretend that happened. So if somebody just very, very softly touches your hand, or somebody squeezing your hand, this is a different stimulus. And even if you close your eyes and somebody gently touches you, you feel it's gentle touch. Or somebody squeezes your arm, even with your eyes closed, you feel it's a lot of strings. That's a stimulus. But your brain receives the same action potential. So how your brain knows the difference? Now, the strong stimuli generate action potential more frequently. So over here, let's look. So if somebody gently touching you over here, it's a stimulus. So here's the frequency of action potentials that your brain receives. Now, if somebody is squeezing your hand, your brain receives this frequency of action potential. And this is how your brain knows that's a soft touch and this is very strong touch. And if somebody touches you and you don't feel it, that means this is sub-threshold stimulus, so no action potential. Because sometimes it can happen, right? Somebody touches you so lightly that you don't feel it at all. Now, that's how your brain determines the strength of the stimulus, right? So if I start talking like this, your brain receives this type of stimulus because this type of action potential when I lower my voice and you can hardly hear me. So that's what your brain receives. And when I start talking very loudly, how I usually do, I'm sorry, that's what your brain receives. And this is how it determines between weak and strong stimulus in the action potential. Okay, now absolute refractory period. So let's look what it is. So that's our action potential. Like in this period of time, doesn't matter how strong the stimulus is, your neuron cannot generate another action potential. This is called absolute refractory period. So no stimulation can cause action potential. Now this area in dark blue is a relative refractory period. So when stimulus is really strong, then neuron can generate additional action potential. Right? That's what absolute and relative refractory period are. So you can read over here. So where is that? Yeah, so you can read this. It's a lot of explanation. I just, I'm giving you the main idea. So just remember that in absolute refractory period, no stimulus, it doesn't matter how strong it is, can generate action potential in relative refractory period. So we're not back to normal yet, to resting potential, but strong stimulus can generate another electrical impulse. Okay, now how fast this electricity moves along your neuron and what affects this velocity. So it's called conduction velocity. What affects conduction velocity are diameter of axon. Larger diameter, the faster this impulse, and effect of myelination. So if you remember from Bio 58, myelin sheaths, it's like a lipid structures that cover axons. So if you have myelinated axon, then conduction is quick. And when it's unmyelinated, then conduction is slow. And when we have myelinated axon, we call this conduction saltatory conduction. So here's effect of myelination. As I told you then, when we have myelinated axons, we call it saltatory conduction, and it's about 30 times faster than in non-myelinated axons, right? In myelinated, it's 30 times faster. So let's look at diagram. So first we see dendrites, right? Dendrites only generate graded potential. So here are stimulus, and this kind of show our electrical current, and you see it decreases with the distance. Now, if we're looking at axons, here's our unmyelinated axon. Now, here's the stimulus, and this would show in white, this is electrical current, and it does decreases a little bit, but because we have voltage-gated channels, we don't have any voltage-gated channels in dendrites. But here, because we do have them, they open along the axons, and sodium moves in, and we kind of return back to the same strengths. of the, oh, I'm sorry, the same speed and strength too, right? So that's unmyelinated. In myelinated, we have those voltage gated channels only at node of Ranvier, and electrical current, it appears kind of like jump from here to here, from here to here. So that would increase the speed of conduction. There is some disorders, autoimmune disorders like multiple sclerosis, that affect this myelin sheath. So when myelin sheath, it get damaged and non-functional because of sclerosis, then it's decrease the speed of conduction, and symptoms include visual disturbance, weakness, loss of muscular control, speech disturbance, urinary incontinence, because your nervous system regulate all of those functions. Right, so this impulse conduction slows and eventually ceases. So that's a multiple sclerosis, autoimmune disease, when your immune system attacks this myelin sheath. And this only happens in central nervous system. We do have similar disease in the peripheral nervous system. It's called Guillain-Barré syndrome, but multiple sclerosis only in central nervous system. And we cannot treat it, but we can hold symptoms at bay, reduce complication, reduce disability with drugs, all right? Capazone, interferons, you don't need to memorize the drugs, but we do have drugs that just decrease your immune response. Now, nerve fibers classification, your nerve fibers, and I'm reminding you that nerve fibers are axons. So they differ in a diameter, right? They can differ in diameter and degree of myelination. And because they differ in diameter and myelination, they differ in speed of conduction. A larger diameter, faster the speed. Myelination, we have fast speed, right? So we can classify fibers into group A, B, and C. And A are the largest, myelinated, B is intermediate diameter, lightly myelinated, and C are smallest diameter, unmyelinated, so fastest, medium speed, and slow speed. And the fastest fibers are actually, in your somatic, sensory, and motor fibers. So somatic sensory means sensory information that you understand. So sensory information that reaches your consciousness. So everything that you understand, what you see, what you hear, what you feel, cold, you know, soft, and what else? You can feel pain and all this stuff, right? So that's your somatic sensory. If pain comes from outside, of course, we have visceral pain as well. And motor fibers, somatic motor fibers that innervate your skeletal muscles. So everything that you can control actually have the fastest fibers. And this group B and C in autonomic nervous system, right? So it's your heart muscles, your smooth muscles, and your visceral sensation, right? So if you have pain from outside, that's very quickly gonna reach your consciousness. If you have something from inside, then it takes a little bit longer to get to your brain. And also it takes longer time for your brain, well, for impulse from your brain, you know, to reach, let's say, your stomach than to reach your skeletal muscles, right? So now, what is synapse? Synapse is a junction that mediates information transfer from one neuron to another neuron or effector cell. So, so here we see the synapse. So synapse is a junction between neuron and effector cells or another neuron. And when we have two neurons, we have presynaptic neuron, conducts impulses towards synapse, and postsynaptic neuron transmit impulses away from synapse. Now we have several types of synapses, exodendritic, exosomatic, exodendritic axons and dendrites, axon and soma, those are common. And less common are exoaxonic, dendrodendritic, dendrosomatic. So we actually have all type of synapses, but those two are most common. And on this diagram, you can just see that neurons doesn't necessarily synapse with another neuron. They can synapse from effector organ, for example, with your blood vessels. Now we have chemical and electrical synapse. Let me just go to this diagram. Now, this is chemical synapse, the one that we, present in your nervous system, right? So that, well, this is also can be present in your brain, but chemical synapse involve a release of neurotransmitters. So we have electrical signal reaches this terminal, right? Axon terminal, and then neurotransmitters are released. And neurotransmitters are chemicals. That's why this is called chemical synapse. This is very common. We do have electrical synapses like in your heart, in your brain, in some embryonic nervous tissue, less common when we don't have any neurotransmitter released. We just have this gap junction and electrical signal just passes from one neuron to another without this neurotransmitter delay. Okay, now synaptic cleft is this fluid filled space that separate presynaptic and postsynaptic neurons. And that this cleft prevents nerve impulses from directly passing from one neuron to the next. Okay, let me see over here. I'm just go to this slide. So here's we add this axosomatic synapse. So axon synapses with soma. So here's axon, right, of one presynaptic neuron. And this is called axon terminal. This is synaptic cleft. And this is soma of the second postsynaptic neuron. Now, when electrical impulse that we call action potential reaches this terminal, remember, when we have action potential, cell inside became now positive instead of negative. So now, instead of being negative inside, this membrane is positive inside. When that happens, voltage-gated calcium channels open. So calcium start moving in. Now, what calcium does, it causes these vesicles, right? And those vesicles with neurotransmitters, so neurotransmitter containing vesicles, to move toward the cleft and fuse with the membrane and release neurotransmitter, right? So action potential reaches terminal, calcium gated, voltage gated channels open, calcium influx, vesicles fuse with membrane, neurotransmitter is released. Now in this neuron, that is postsynaptic neuron, on a membrane, we have receptors for those neurotransmitters. Those are ion channels, and those are ligand gated or chemically gated ion channels. So you see over here, neurotransmitter attached to the channel and open the channel, and sodium start moving in, and we create graded potential. So again, graded potential, and if it's strong enough, we will have action potential in this neuron. Right now, what's gonna happen with this neurotransmitter, it's either can be reuptaken back, so it can go back to the presynaptic neuron, it can diffuse away from this cleft, or it can be degraded by enzyme. So, it doesn't stay in a cleft for prolonged time. So that's not good for nervous system. Right, so here termination of neurotransmitter is a degradation by enzyme, reuptake by axon or even astrocytes can take in those neurotransmitter, or they can just diffuse away from synaptic cleft. Now, neurotransmitter must be released, diffuses across the synapse, bind to receptor. This is called synaptic delay. So it's a time needed to do all this stuff. And synaptic delay is a rate limiting step in neuron transmission. Now, postsynaptic potential. So where's our postsynaptic potential? So postsynaptic potential happens in postsynaptic neuron over here. So it's a graded potential that happen now in here in the soma of this postsynaptic neuron. So those postsynaptic potential, they can be either excitatory or inhibitory. So EPSP or IPSP. Now, it's excitatory if it moving toward the 0, right? Then it's called EPSP. And usually this happens because sodium channels open. It can be potassium as well, but let's just think about sodium. If sodium start moving in, right? Sodium start moving in, of course, we're moving towards 0. So less negative. So that's our excitatory postsynaptic potential. Well, not always sodium. What if chlorides start moving in, right? If chlorides start moving in, it's negative. Then we're moving away from 0. And this is called inhibitory postsynaptic potential. Because your neurons, they can really, their activity can be regulated. They release inhibitory and excitatory neurotransmitters, right? So depends. So either you want to stimulate your effector cells or you want to inhibit it. Now we have EPSP and IPSP. A single EPSP cannot induce action potential. They summate. IPSP also can summate with EPSP, and then they can cancel each other. So let's see what happens. So for example, here's my postsynaptic neuron. That's postsynaptic neuron. Now you see this kind of like greenish stuff. This is presynaptic neuron. So this neuron send excitatory, right, stimulus to this neuron. So we have this one synapse. But what happens if over here in the same synapse, we just we increase the frequency of the action potential, right? We call it fire. So this axon fire, but this axon fire in a rapid order. So if it's just, it's fire one graded potential, another graded potential, but both of them are not strong enough to generate action potential, sub-threshold. But if it's fire more rapidly, they can add, right? And it's called temporal summation. Or what can happen, so here's one axon, right? And we gonna have another neuron that also fire. So here we have two synapses. But because we fire both synapses, they also add up and it's called spatial summation. Or we have excitatory and inhibitory postsynaptic potentials, and they can cancel each other, right? So, okay, now very quickly about neurotransmitters. Most neurons make 2 or more neurotransmitters, and we know about 50 neurotransmitters. But definitely we have way more, and they can be classified by chemical structure and by function. So by chemical structure, what you guys, good for you to remember, acetylcholine, that's very common neurotransmitter. First, it's released at neuromuscular junction. And this is what you cover in 58, right? So where your neurons innervate your skeletal muscles, acetylcholine is released. It's synthesized by enzyme chlorine acetyltransferase and degraded by enzyme acetylcholine esterase. Acetylcholine esterase, right? So that's very common acetylcholine, and its classification others, really, like others. Then we have biogenic amines. Biogenic amines, that means they have this amino group. NH2, you see this NH2? So those are biogenic amines, and they include catecholamines, very famous, dopamine, epinephrine, norepinephrine, and also serotonin, histamine. So many neurotransmitters are biogenic amines, and they have this amino group. So broadly distributed in the brain, plays role in emotional behavior and biological clock. We also have amino acids. So before we talk about biological amines, only amino group, amino acid includes amino and carboxyl group. This is COOH group. So if COOH, so that's carbon over here. So this is amino and a carboxyl group, and it's make it amino acid. Also very famous GABA, inhibitory, glycine, aspartate, glutamate, right? So that's amino acids, neurotransmitters. Peptides. Well, peptides are short proteins. Substance P, for example. This allow you to feel pain. Endorphins. Endorphins reduce your pain. And some peptides include gut-brain peptides, so that affect your digestive system. Another group are actually purines or ATP. ATP can be a neurotransmitter, and it's act in both central and peripheral nervous system. Can produce fast, slow response, induce calcium influx in astrocytes, and provoke pain sensation. ATP is actually inflammatory chemical, so it's make you feel pain, similar, and substance P as well. So this inflammatory chemical allow you to feel pain, and ATP as well. Endorphins reduce your pain. Okay. And of course, we can also classify neurotransmitter based on their function. So before was based on the chemical structure, but based on the function, we have either excitatory or inhibitory. And this depends not on neurotransmitter really, but on receptor. So remember that GABA and glycine are usually inhibitory, glutamate are usually excitatory, and acetylcholine, very interesting. It's excitatory in neuromuscular junction in skeletal muscles, so it excites your skeletal muscles, but it inhibits your cardiac muscle. Now, neurotransmitter action, it can be direct and indirect. And when it's direct, then response is very rapid. And when it's indirect, then it's long-lasting effect, but not that rapid, right? So makes sense. Now let's look over here at the direct action and indirect action. Now, so if we have direct action, neurotransmitter attaches to ion channel and opens this ion channel. It does has a receptor over here, and this receptor is called ionotropic receptor, right? So this is direct action. Neurotransmitter attached to ion channel and open ion channel. Well, if it's indirect action, neurotransmitter attaches to receptor, but then it activates the receptor, but it takes several steps before ion channel is open, right? And then this receptor called metabotropic receptor. Tropic means you feed something, like you eat, right? Ionotropic, kind of like it feed on ions, and metabotropic, it feed on chemical reaction, right? So it is a ionotropic receptor when it's direct action, or metabotropic receptor when it's indirect action. And this takes longer, right, to have a response, because we have several steps before we open ion channel. Now, in receptors, I just told you, either we can have ionotropic receptor, shown over here, or metabotropic receptor. Let me show you. That's why this one, right? So this is metabotropic receptor. So neurotransmitter is attaches to its receptor, but here's where our ion proteins. So let's imagine the goal is to open this protein, but receptors are way, away from this ion channel. Now, when this happened, right, we call it metabotropic. Oops, let me just go back and just remind you what we're talking about. We're talking about neurotransmitters, right? And neurotransmitters can either have direct or indirect action. If they have direct action, that their receptors are called ionotropic receptors. If they have indirect action, their receptors are called metabotropic receptors. Now, in a ionotropic, it's kind of easy, right? Neurotransmitter attaches to ion channel right over here, and channel opens, ions moving in. Now, when we have metabotropic, this is what happens. So let me make it bigger. Neurotransmitter then called first messenger. So neurotransmitter binds to the receptor. It activates this receptor. Receptor activates another protein. Most commonly, it's a G protein. So that's why it's called G protein linked receptor. So that's a receptor, but it's linked to G protein. So when neurotransmitter attaches, it activates this G protein. Now G protein, right, it's kind of and it's active now, so it start moving along the cell membrane and it's activate another protein. This is enzyme called adenylate cyclase. So this enzyme gonna convert ATP into CAMP. So it's a molecule that is made from ATP, and CAMP is called second messenger. Now this second messenger now can open ion channels. So first messenger neurotransmitter does not open ion channel. First messenger causes those reaction and activation of second messenger, and then second messenger open ion channels. But second messenger can also do other stuff. It can maybe activate other enzymes, or it can even move inside the nucleus and affect DNA, right? So that's how metabotropic or indirect action of neurotransmitters happen. And we're almost done. We're not that bad, only hour and a half for this lecture. So neurons never function on its own. They form functional groups, right? So what are those functional groups? We call them pools, right? So we have simple neuronal pool. And this is how it looks like. We have one presynaptic neuron, then its branches, right? So that's axon, those are axon terminal. So it's branches and it's synapses with several postsynaptic neurons. So this is simple neural pool, simple neuronal pool. And we have discharge zone where we have, most of neurotransmitter released, right? And we have facilitated zone. Now, when it's not simple pool, then we call it circuits. So we have several types of circuits. So here's diverging circuit. Diverging means we have one input and several outputs, right? And so several outputs can go in one direction or in different direction, but it's kind of like increasing, we have ever increasing number of neurons, right? So that's a neuronal pool and it's diverging circuit. So it's allow you actually to amplify your, electrical signal, right? So you amplify it. You have one input and several outputs. Convergent circuit, when we have, it's opposite of diverging, and we have several inputs and only one output, right? Or we can have even several inputs from one neuron, but we still have only one output. So this explains how different sensory stimuli can elicit the same memory, right? So either you have different stimuli, but you have the same memory of those smell, for example, right? Now, reverberating circuit, we have a chain of neurons, and some of them have these collateral synapses, so they pretty much electrical signal. We have one input, one output, but electrical signal kind of making this circuit. It can just go around the circuit and then output. And this circuit involved in a transmission of information such as when you're walking or any repetitive activities, breathing, sleep-wake cycle, right? So that's kind of like it's go through the circuit, so that's good for repetitive motion. And the last one is parallel after discharge circuit. We have one input, one output, but in between we have kind of like electrical signal go through the parallel, okay, simulate several neurons in parallel array. So we have this parallel array. And what is interesting over here, we have one input, one output, but the signal reaches this neuron at a different time. So it reaches signal faster if it goes this way, because we have, just this way, we have only one synapse. Here we have two synapses, right? This is the most complicated, and it's involved in your mental processes, such as, let's say, mathematical calculation. So the point is you never have like two single neurons going from your finger to your brain, right? One neuron in your finger, one neuron in your spinal cord, and one neuron in your brain. We have pools of neurons, and it can either be a simple pool, or it can form different circuits, right? Like diverging, converging, reverberating, or parallel after discharge, right? So that's it for this chapter 11. It's a lot, but the good thing is you can rewatch this video if you feel like you missed some information. So I'm gonna stop recording.